Hi doctor, my name is XIN X69910. Today I will present the subject of 3454 Aquaculture Biotechnology. The topic is application of molecular marker in fish for genetic improvement in aquaculture. The first is introduction. The application of molecular marker in aquaculture has become an essential tool for enhancing fish genetic improvement. This biotechnological method enables researchers to identify and select fish with desirable traits such as disease-resistant, fast growth, and environmental tolerance. Techniques like macrosatellite, which is SSR, single nucleotide polymorphic SNP, and quantity track logic, which is QTL, mapping has been widely used in various fish species including tilapia, carp, and salmon. These experiences help improve breeding efficiency and sustainability, supporting the long-term productivity of aquaculture system. I will go through the application of molecular marker in aquaculture. The first is molecular markers such as SSRS, AMPS, and QTLS are powerful tools used for genetic improvement in aquaculture. Their application include genes mapping, genetic diversity analysis, disease-resistant breeding, and traceability of aquatic species. I also include the table to show that the molecular marker can use in genetic diversity, DNA fingerprinting, prevent like identification, hybrid purified genes parameting, and the genes mapping. This is common molecular, which have three types. The first is microsatellite, which is SSR. They are short repeating DNA sequence. They are widely used in fish genetics to assess genetic diversity, identify traits, and support selective breeding. The second type is single nucleotide polymerase SNPS. They are DNA sequence variations that occur when a single nucleotide, which is A, T, C, or G in the genus, is altered at the specific position among individuals or of species. The third is quantitative trace logic mapping, which is QTL. So it is a method used to find specific part of the genome linked to important traits like growth, disease resistance, or body size. In fish, it compares DNA and trait data from offspring to locate this resident for selective breeding. So we will go to the first microsatellite, which is SSR. This is the first result and the first journal that I find. The topic is microsatellite marker, what they mean and why they are so useful. So the first point is microsatellite are short, repeating, and secure. The second is they show high genetic variation, polymerase, and the third is they are useful for studying genetic diversity, and the fourth is they help in mapping genes and QTLS, and the last is they are used in breeding and parents analysis. So we will go to the second journal, which is what are microsatellite and how to choose the best tool, a user-friendly review of SSR and 74 SSR mean tool. The first is SSR and SMP marker help study fish genetics. The second point is SNP arrange improve selection of growth rate, and the third is marker support sex identification in fish, and the fourth point is used to track fish population for conservation, and the last point is support marker assist breeding in aquaculture. So we will go to the second, which is single nucleotide polymorphic, which is ANPS. So this is the first journal of my result of single nucleotide polymerase. The topic is single nucleotide polymerase marker with application in conservation and reproduction of aquatic natural population. So the first point is SNPs are useful for fish genetic improvement. Second is they help study population structure and diversity. SNPs can identify farm vs wild fish. And the fourth is they detect threats like disease resistant. And the last is useful for selective breeding and conservation. We will go to the second journal of my result, single nucleotide polymerase. The topic is an array of hope for genetic improvement of aquatic species and fishery management. SNP arrays detect genetic difference at single DNA bases, and the second is they help find marker linked to fish growth and health. And the third is used in genome-wide study to locate useful traits. And the fourth is improve accuracy in selective breeding program. So the last is useful for managing genetic diversity in aquaculture. So the third is quantitative trait logic, which is QTL mapping. This is the first journal of my result for quantitative trait logic QTL mapping. The topic is QTL mapping designed for aquaculture. So the first point is QTL mapping 5 genes linked to fish growth. Second point is it uses SSR marker to build linkage map. And then third is help identify fast-growing fish family. And the fourth is support genetic improvement in aquaculture. And the last point is useful for selective breeding program. I will go through the comparison of molecular marker in aquaculture. The first one is SSR. This is best for genetic diversity parameters, and the SNPs is best for high throughput trait screening, and the QTL mapping is best for bidding for complete traits. And the second is pros. SSR is cheap and highly variable, and the SNPs is pricey and the gene-wide coverage, and the QTL mapping is linked genes to trait daily. And the third is count. SSR is limiting scalability, and the SNPs is expensive technology, and the QTL mapping is need large datasets. And the last is example for use. If for SSR used to tilapia pedigree tracking, and the SNPs we use for salmon growth selection, and the QTL mapping we use for carp disease resistant. So if you want to simple and low cost, we can use the SSR, but we want to precision and the scalability, we can use to SNP. And the last is if we targeting multiple traits, we can use the QTL. So I will move on to the discussion. The use of molecular markers such as macrosatellites, SSRS, and single nucleotide polymerase SNPs has greatly enhanced genetic improvement strategy in aquaculture. SSRS, due to their high polymerase, are useful for analysis, genetic diversity, and cross-housing linkage made, while SNPs offer higher resolution for identifying traits like growth rate and disease resistance. Recent developments such as functional gene-based SSR marker in red tilapia had enabled researchers to link specific genetic regions to important aquaculture trends, supporting more accurate selective breeding programs and ensuring long-term genetic sustainability. In conclusion, molecular markers like microsatellite SSRS and SNPs have transformed genetic improvement in aquaculture by enabling trait selection for growth and disease resistance. Their application, like in species like lake tilapia and geohead seaborn, show promise for sustainable breeding, Future integration with genome-war study will further boost aquaculture productivity and efficiency. This is the reference. Thank you, Doctor.